Hello students, today we are going to discuss about a new topic, that is genetic code. Before that, in last class we have discussed about transcription, where the mRNA was synthesized, and you have seen already the process of RNA processing or RNA modification. Okay, one thing I want to discuss about the previous class was about the mature RNA. Okay, that is your transcript RNA. After transcript RNA, you get your mature RNA. But there's one important point that I forgot to not explain was UTR, that is untranslated region. Now this is your, it was capping, where you have capping was done, that is 5 prime, and it was methyl guanosine triphosphate. After modification, okay, then you got your, this is your coding region, then you got your poly A tail at the 3 prime. Okay, now this is your mRNA or mature RNA. mRNA. Now you know that there is a start codon, right? This is your O, for example. I'll discuss about this. O. This is your start codon. Start or the initiation point. Okay, start codon. Now this one is your termination codon. Termination codon. So what is untranslated region? Untranslated region is the one which will be present at the 5 prime end, okay, before the start codon, and after at the 3 prime end after the termination codon. Okay, this question was asked in NEET 2018. Okay, so I will consider this as UTR, untranslated region or non-coding region. Okay, now in the next topic we are going to discuss about the genetic code. Now in this there is a scientist called as George Gamow. Okay, now he was thinking that he thinks that there are only 4 nitrogenous bases, right? 4 nitrogenous bases. For DNA, that is A, T, G, C. Since we are talking about mRNA, so it will be A, U, G, C. Fine. So in mRNA, those nitrogenous bases A, U, G, C are present. Okay. Just say that this is 5 prime to 3 prime, and there is a nitrogenous base A, U, G, C, A, U, G, C. Now, naturally there are only 20 amino acids. Okay. First point. Second point is 20 amino acids. I hope you know what are amino acids. Okay. Amino acids are the building blocks of protein. It means that proteins are made up of amino acids. Now, if you understand genetic code properly, then only you can understand how proteins are synthesized from mRNA. Okay, so out of these 20 amino acids, there are these are naturally amino acids, but even other amino acids are also synthesized. Okay, are also present. Okay, but for all those 20 amino acids, he says that it has to be coded. Coded means it has to be coded by 3 nucleotides. He says that it has to be coded by 3 nucleotides. What is that 3 nucleotide? For example, AUG. For example, U So 3 nitrogenous base, okay, which will code for 1 amino acids. For example, 1 amino acids, just a leucine, okay. Another amino acids, for example, AA2, okay. For example, phenyl, PHE, phenylalanine, okay. So for 3 nitrogenous bases, it will be coded for 1 amino acids. So he said that for all the 20 amino acids, okay, there will be 64 codons. So he used a formula with the help of permutation and combination. Okay, he gave a calculation of, that is 4:4 nitrogenous base and 3 nucleotides. Okay, which will give you 64 codons. Okay, but only 61 will code for 20 amino acids. 61 will code for 20 amino acids. Remaining 3, okay, 61, 62, 64, right? So remaining 3 will not code, okay, will not code for any amino acids. So there were other scientists, okay, in order to support the George Gamow, there were 3 scientists or 3 experiments, okay, as I say, there was 3 experiments which helped, okay, to prove that yes, the genetic code, okay, the genetic code is of 64 codon. The first experiment was done by Hargobind Khorana, okay, Hargobind Khorana, okay, Hargobind Khorana. Now this scientist, he got a Nobel Prize for synthesizing the RNA molecule. Means these scientists have developed RNA molecule, not naturally, artificially, okay. In his lab, he had developed this RNA molecule with the help, okay, of chemical method. Artificially, okay, by chemical method, he had developed the RNA molecule. The second was, okay, Nirenberg, okay, N-I-R, Marshall Nirenberg. Okay, Marshall Nirenberg. Now this, he had developed a free living system, okay, free living system for protein synthesis. Another very important enzyme, that is Severo-Ocha enzyme, okay? Severo-Ocha enzyme. Now, this Severo-Ocha, okay? Severo-Ocha enzyme. This enzyme is help, okay? This will be helping for the polymerizing process of the RNA. Helps in polymerizing RNA. Okay. So this enzyme is also known as polynucleotide polymerase. Okay. Polynucleotide phosphorylase. Polynucleotide phosphorylase. Okay. So these are the 3 things, 3 important experiments which was conducted by different scientists, and then the idea, which was the idea of the George Gamow, okay, was supported, and a new code or a new chart has been drawn out. Okay, and how to make this genetic code, now we are going to discuss that. So first of all, you make a 4 columns, 1 2 3 4, 1 2 3 4. Now based on this chart, you have to do an assignment. Okay, so listen properly. First you have to write U, pyramiding. Okay, U A U C, then A G. So above U C A G. So U C A G. So I'll make 2 columns. Okay, and then you have to make remaining. Okay, so this one is, first of all you have to see the first point. Okay, this one, then first write 1 2 3 4, 4 times. Okay, then this one 1 2 3 4, and the U C A G. Okay, once more, this one, this point, the first point, so U 1 2 3 4. Okay, the middle one will be this position, so 1 2 3 4, and the ending will be U C A G. Okay, same as like that here. It will be, since it is C, so first you have to make C, 1 2 3 4. Okay, and then middle one is U. U is uracil. Okay, then you have U C A G. Okay, same as like that in this point. Go for A, then 1 2 3 4. Then the middle one is U U U U. The last point is that side. It is U C A G. Same thing here. G 2 3 4. Uracil, uracil, uracil. Then U A U C, right? U C A G. Okay, same in this column also. You are going to fill up the same way. Okay, this will be your assignment to make it on a big chart paper. Okay. Now, what is this? If you count it in each boxes, you'll get 4. Okay, say 1 2 3 4 4. So if you count it all these, you will get 64 columns. It means that, let me give you a simple trick. Okay, each, see, there is only 3 nitrogenous base. 3 nitrogenous base will code for 1 amino acids. Okay, for these 3, 1 amino acids. For these 3, 1 amino acids. Okay, so now we're going to discuss about the silent features of the genetic code. Okay. Just right here. The first point, triplet. It means that your genetic code is triplet. Why triplet? Because there is only 3 nitrogenous base. See, C U U. It means that it is triplet in nitrogenous base. Okay, and 61 will code for 20 amino acids, and 3 will be stop codon. Okay, stop codon. Now, where will be the stop codon? Here. Okay. Actually, the stop codon remain in this position. Okay, let me draw once. This is U, so U 1 2 3 4. Okay, then A 1 2 3 4, and then U C A G. Okay, which one is a stop codon here? These 2 will code for different amino acids. Okay, I'll give you an idea. You look in your textbook, it is here, and this UAA. This is a stop codon. It does not code for any amino acids. So we also call as termination codon. Termination codon. So this is both UAA and UAG. These two are termination codon. There are 3 termination codon, so you may be thinking, where is the another one? Okay, another one will be here. Okay, so it will be U 1, 2, 3, 4, A, okay, then U, C, A, U, C, A, G, okay, A. So this is the termination codon. Again, fine. Sorry, see this G, okay, G, 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 G. This one will be G, okay. So another termination codon is present here, fine. So that's why we have 3 stop codon and 61 amino acids, which it represent your 20 amino acids. Now, point number 2 is, it is, first is triplet, second is the comma-less. So you don't see any, if I write A, U, G and comma, U, so this is wrong. So it means that there is no punctuation. Okay. Number 3 is degenerate. Degenerate means, when I say degenerate means, just say for one amino acids, for one codon, one codon. See, there are so many codon. There are 64 codon. Okay, for one codon. Okay. Not one codon, for some amino acids. Okay, more than one codon. I'll give you an example. Just say that I'll take one amino acids. Okay, this is for leucine. Okay, Leucine, leucine. You can see that there are 4 codon. Okay, here also it is leucine. Okay, these 2 and these 4 is for leucine. So even for this leucine, you can see 1, 2, 3, 4, 5, 6 codon. It means that for one amino acids there is more than one codon, so that is said to be degenerate. The last important point is universal. Universal. Okay, so universal means, for example, from human to bacteria. So if there is a UUU code for phenylalanine. So if UUU nitrogenous base is there, even in bacteria or human being, this code will be same. It will code only for phenylalanine. Okay, so these are the 4 important points of the genetic code. In the next topic, next class, we are going to discuss about the structure of tRNA.